Researchers have taken the tiny human brains we can grow from stem cells, implanted a brain chip into them, and then implanted them into that brain organoid in the head of a mouse. Let's talk about it. If you're not familiar with our tiny human brain friends, they have a number of uses, including doing computation. But I think the more compelling work is in recovery from brain injury. There's been some research in the use of them to recover from strokes, and it is promising, but there's a few things that need to be worked out before we put them in humans. When growing a disembodied human brain, the lack of a body causes some issues. It does end up suffering from hypoxia after a fairly short period of time. There has been some efforts to give them a circulatory system and change their environment, and some have been able to keep them alive for 10 months or more. This makes them particularly helpful for computation. If you're going to teach a tiny human brain to do computation for you, you want it to remain alive for a while. In the process of learning how to make it do computation, it has been noticed that it does better if it has something to do. A disembodied brain needs something to stimulate it. That's where using brain-computer interface comes into play. You can take that semi-mature brain organoid, attach it into a computer interface, and then implant it, but continue to control it after it's there. Doing so keeps the cells activated, it keeps it doing stuff, and it allows it to reconnect with the other brain structures quite well. Brain organoids are very invasive. You can take a human brain organoid, put it in that mouse's brain, and it'll make connections not just with other neurons, but all the way down to the spinal cord. They're so good at doing this that you can even take a decapitated fetal mouse and it can grow projections into the spinal cord and even take control of those muscles. The specific reason they use human brain organoids, even with things like mice and monkeys, is because if it is ever to be applicable to human medicine, they're going to have to work with our brains. There also seems to be the issue that mouse brains tend to overgrow. So if you were to implant them into a damaged portion of the brain, it may become too large and cause pressure on the brain and further damage. This one is really cool because it has several applications. It could be the future of attaching brain-computer interfaces to your brain. You may want to make a copy of your brain, a tiny one, attach that interface, and then implant it, preserving your structures. But time will tell on that. This research does have a hell of a lot of promise. There's evidence that you can repair portions of the brain that are damaged that cause blindness or loss of other sensations like smell. The reason that we haven't made the jump to people just yet, and I wouldn't doubt that somebody's asked a researcher to do it, the reason we haven't jumped to people just yet is because we need to make sure that we have the best methods possible before we make that jump. If you do it too quickly, it can end up setting the field back, even decades. When researchers first made the jump to mice and rats and even monkeys, they didn't have the best results. They didn't know that there was a period that they should wait before implanting them, and they didn't know that the brain organoids did better after they've been grown for a while. This is likely one of the major problems with trying to stem cell seed a brain to heal an injury. It doesn't work, the cells don't survive. Knowing that they do better when they've had electrical stimulation, knowing that they do even better if they have that electrical stimulation while still in the skull of the patient. There may be more advances down the line, and getting treatment too early can mean the end of the patient, the end of the research, and not having the opportunity to have that treatment down the line. It is frustrating, but these are all things to consider. I'm excited for where this field is going.
Complex than the tiny prokaryotes swarming in the culture.